Hello everybody. I'm happy to see all of you and starting the new year with my talk. So what I'm going to talk is as you can see, the title is the topic about entanglements in RNA 3D structures. And the topic itself is connected with the topology of RNA 3D structures. So in general, topology is kind of mysterious area of mathematics. But what I'll be talking about is the pattern of passages of RNA strand. If I talk about topology, if I think about topology, I try to answer several questions, like, for example, which parts of RNA are connected to which. So I somehow distinguish different parts or motifs or regions in RNA structure and then try to find out how they are connected, how they are positions relative to one another in space, okay? Another question, which is directly connected to my talk, is whether the elements of the structure form some crossings in space, or whether they do create some loops or entanglements. Next, I try to answer the question whether the chain, the RNA chain, is somehow knotted. So does it include any topological knots, or is it free of such structures? And finally, how all the elements like helices, loops, junctions wave through each other. So all that, all those questions are related to what I think about when I talk about topology of RNA structure. Now what you can see on this picture, on this slide, is in the middle the secondary structure of RNA. Some RNA, short RNA chain, encoded in the bracket notation. And on both sides, there are two 3D structures, 3D models of the structure that were modeled by some predictive method. Starting from the secondary structure we can see in the middle, okay? And as you can see, they differ, okay? They differ quite in quite an observable way. So, is anybody can say that one of these models is correct and the other is not? And maybe can we somehow assess whether one of them is biological reality, is possible in nature or not? What you can probably see here is that one of these structures, one of these conformation is a knotted conformation. This is the one on the right side, on the top. The other is not. And that's the difference in terms of topology. So this is the main difference I want you to pay attention to. So does RNA topology matter? Of course, the answer is yes. Yes, so we can predict the structure with different topologies. Some of these structures, some of these topologies may contain some artifacts, like some spurious knots, entanglements, or some distortions. And what I want to answer with my talk is how can we automatically detect and fix such topological anomalies if we consider them anomalies. And what I want to tell you also that most predictive methods, including the newest ones, the recent ones, predict structures with some hidden artifacts, although most of them do not validate the topology of the predictions. So as a result, we can obtain some models that maybe do not reflect biological reality. Okay, so they may be somehow misleading in terms of biology, function, and structure itself. Okay. When I'm looking at a structure and trying to assess, to evaluate the topology, I can look at it from global perspective or from a local perspective. When we look at the structure from global perspective, we can try to find out whether the structure contains any topological knots. Okay, so knots occur when the backbone of RNA chain becomes somehow permanently entangled in such a way that it will not disentangle itself during some moves, some dynamics. So it forms some loops that cannot be untangled without breaking the chain. We think that, or it's usual, most people think that such knots, such topological knots, are computational artifacts in the models that we build in silico. We can also look at the structure locally and try to find whether there are any elements, regions of the structure of the chain that form entanglements. When we look at the structure from the local perspective, we look at the elements like loops, helices, single-stranded fragments of the chain, and we try to find out whether there are some collisions between these elements. So when I will talk about entanglements of structure elements in this talk, I will consider this local perspective. So I will look at the structure just locally, focusing on some structural elements. Okay, and the work that we've performed in our team, we used to treat RNA structure as a kind of assembly of structural elements. And since the very beginning of our work on modeling RNA 3D structure, we treated the structure as kind of assembly of elements, and that's how our system RNA Composer works when it composes the 3D structure. It exactly assembles the structure, the entire structure from structure elements. So when we started to look into the topology of the structure, we also followed the same idea, the same approach. And we started to look into the structure as an assembly of structural elements. And we decided to find out whether the elements we can distinguish in a structure can form entanglements and occur in a kind of collision in the 3D space. So what we did first was we defined theoretical types of entanglements of structural elements that we potentially could find in predicted RNA 3D structures. What you can see here in the picture are 9 types of entanglements that we distinguished. So each entanglement is composed of at least one closed element. By a closed element, we mean a loop or dinucleotide step. And the second element is either an open element, like a single-stranded fragment, or another closed element. And the symbols here, which you can see under each of these types, tells us what types of elements are entangled here. So we have three types of links, or we can call them interlaces. We can see that all these types involve two closed elements, so either loops, which are denoted by L, or dinucleotide steps, which are denoted by D. So we have L and L, D and L, and D and D type of link or interlace. And we have 6 types of lassos. In these types of entanglements, we have one element which functions as a kind of lasso, which is a lassoing element, and the other element which goes through the lasso. And again, the lassoing element is a closed element, like a loop or a dinucleotide step, and the one that goes through the lasso can be either an open element, so single-stranded fragment denoted here by S, or a closed element denoted by L, loop, or the dinucleotide step. Okay, once we define those theoretical types, we ask a question whether all of these types are possible, where they can form in nature, in biological molecules, and maybe some of them are misfolds. So what we think is that all these 3 types of links, interlaces, are misfolds. And also deep lassos are misfolds, while shallow lassos may form in nature. What is the depth of a lasso? So we decided to compute how many nucleotides goes through the lasso, and this number of nucleotides is the depth of a lasso. Okay, so we just compute the number of nucleotides that goes through, and we say that if this number is less than 5 nucleotides, we can say, okay, the depth is this is the shallow lasso. And if the depth is over 5 nucleotides, then we say this is the deep lasso, and it cannot disentangle itself spontaneously. So probably the deep lassos are also misfolds, the same as links. Okay. Then we implemented a procedure that can detect entanglements of structure elements in an automated way. And this is how it goes, how it proceeds. So what we do first with a model, with a 3D model we have, we first coarse-grained the model, and we select pseudo-atoms and atoms in the structure, and we build the polygon chain going through these atoms and pseudo-atoms. Then we obtain polygons, these closed polygons, and we triangle each closed polygon, each polygon actually, and we run the procedure, the algorithm is called Meller algorithm, that searches for punctures in a polygon. And then whenever we find the polygon that is puncture, we say, okay, there is a collision. So there should be some entanglement. And we try to find out what type of entanglement it is. So we go back again to the structure, to our initial structure. We find out, we annotate the secondary structure, and we find out which elements of the structure form the puncture or to form entanglement. And in such a way, we classify the entanglement into one of these types of interlaces or lassos. The algorithm has been implemented in our web server tool, which is called RNAspider. So the tool is available freely at its link, which is provided here. So whenever you have a structure, a 3D structure model, which you would like to check for entanglements of structure elements, you can upload it to RNAspider, run the spider, and find out whether there are any entanglements. So the tool will show you in a 2D and 3D view whether there are any entanglements, and if the tool detects any entanglements, it will show you also what types of entanglements it found and where they are. Okay, now let's go to the global perspective, meaning topological knots. So let's have a look at a whole structure, at a whole chain, and try to find out whether the chain knots or not. To start with, let me tell you a little bit about topological knots. From the point of view of mathematics, a topological knot is a closed curve embedded in a 3D space. We usually look at it by projecting it on a plane and find in such a way that we just find a visualization like the one you can see here on those pictures. And then we try to find out how many crossings we can see on this projection. And the number of crossings here determines the complexity of a topological knot. The crossing number is so-called genus of the topological knot. And we can use, for example, Alexander Briggs notation to denote the type of a knot in such a way that we write the genus of a knot, like for example here, and with this knot we can see on the right side, this is a knot, the 7-2 knot, meaning that the 7 is the genus crossing number, since we have 7 crossings here, and the 2 is a kind of braid number. So we can have at least 2 different types of topological knots with a crossing number 7. You can see here also on a slide an unknot. That is a simple closed curve without any crossings. So we can also say it is 0-1 knot. And the simplest topological knots, knot with more with a positive number of crossings is trefoil. So this is 3-1 topological knot on the left side here. Okay. What you can see here on this slide is the topological knot, which was the first knot I saw in a model that was predicted by our system, by RNA Composer. I mean, the first RNA knot which I saw. One of our users just ran RNA Composer and for some secondary structure, and the system produced such a structure. So what you can see here is a figure 8 knot, also known as Listing's knot. It's a knot with a crossing number 4. So we have 4 crossings when we project this knot onto the plane. And on the right side here down, you can see the diagram of this knot. And as you can see, the diagram is a closed curve. So this is what I told you about topological knots. So topological knots are closed curves in 3D space. So they are not open like the one we can see here in the center, okay, like the knotted structure. So what's going on? What's going on here with this open and closed curves? And what you can see here on this in this RNA structure, which I say it's a knotted structure, you can see it's not a closed curve. Okay. Well, so that's the one of the things that we try to deal with when we look for topological knots in biological structures. We try to close the structures if they are not closed by themselves. And we simulate closings to find out whether the structure, whether the chain is knotted or not. So that's how we actually look for topological knots in biological structures. And that's the simplified diagram of the procedure that is followed in Topoli, which is a Python package to analyze topology of polymers, including RNAs. So what the algorithm there does is it tries to first position the molecule within a sphere. Then it tries to, in such a way that the ends of a chain are positioned on the surface of the sphere. Then we simulate the closing of the chain in such a way that the closing is positioned on the sphere. So we simulate the closing. After the simulation, we compute so-called Alexander polynomial, and the polynomial actually tells us whether the knot exists in this chain or not. We repeat this procedure many times. Here we decided to do it 200 times. In each iteration, we close the chain in a different way. So we just simulate the closing kind of randomly, and each time we try to find out whether the knot exists in a structure or not. And if you find out that over 50% of the closures knot exists in the structure, we say, okay, we consider that this RNA is knotted. So using this procedure, we decided to scan the data sets of predicted as well as experimental 3D RNA structures, and to find out whether there are any topological knots. We also used our RNAspider algorithm for the same data sets and trying to find whether there are any entanglements of structure elements there. We also try to answer the question if there is any relationship between topological knots and entanglements. Okay, so regarding these four sets, you can see here, in one of them, which is a CASP16 data set, we had the target structure with a lasso. So at least here, we have one target that was entangled, or at least it was supposed to be entangled. And all the other subset with predictions, like RNA puzzle subset and CASP15 subset, all the target structures of reference structures were entangled. Okay, And let me show you a few examples of predictions with knots. So first, let's look at topological knots detected with this Topological Python package. So here we have the target structure, which was the largest target in CASP15. And here is one of the predictions obtained. So you can see here that, well, from the prediction itself, the model that you can see here, we can see nothing. But when we try to simplify the chain, the layout of the chain, we found out that there is a trefoil knot there in the structure. Here is another prediction of the same target structure with more complex knot. This is 7-2 topological knot. Okay, and what we asked here was not just about the topological knots, but also whether any of the evaluation measures that are used to evaluate the structure can tell us anything about these artifacts in topology of the predictions. So for both structures, we can see that the RMSD is quite large, although this is also a large structure, so maybe it's not that meaningful here. But we found that TM score is also quite small. So TM score is a little bit connected to the topology of the structure. So here, these values of TM score show us that there is something wrong with the topology. As for anything else that we checked, like for example, interaction network fidelity or clash score, it was not meaningful at all here in terms of topological knot, because here in both cases, these measures were quite good. So interaction network fidelity is here quite high for both models. So the secondary structure is quite well predicted or resembles quite well the target structure, and there are not many clashes here in these structures. Okay, here are some other examples, even more complex topological knots predicted or found in the predicted structures in CASP competition. So there is another prediction for the same target, is over 700 nucleotide long structure. This model here includes even more complex knot with 12 crossings. And here are two other predictions with some other topological knots. These predictions are for another target, which was smaller, but still the predictions contained quite complex topological knots. What I wrote here also is about what was, let's say, the approach behind prediction. So we tried to find out whether groups that predicted and submitted these models used machine learning or not. We just wanted to find out whether there is any tendency in maybe machine learning or non-machine learning methods to predict knotted structures or unknotted structures. So that's what I wanted to show here. Okay, that human groups using machine learning and using non-machine learning methods still can predict quite structures with quite complex topological knots. There are another examples of predictions provided by AlphaFold 3. In this case, we looked into the structure from local perspective, and we tried to find out whether these predictions contain any entanglements of structure elements, so any collided structure elements. And what we found was that AlphaFold can predict structures with entanglements. And it's not just one entanglement, it can be quite many in one structure, especially if the structure is quite large, like for example here. Although there is no direct relationship between entanglements and the size of a structure. So you can see that we can have quite a large structure, like here, for example, a structure on the right side with over 700 nucleotide and having just one lasso. So not a big problem, not a big issue. And we can have a structure which has like 500 nucleotides, but it's got quite a lot of entanglements of different types. So lassos and interlaces of any type. Okay, and here are some aggregated results that show us how we the results of the search for entanglements of structure elements in all of the four sets of structures. So we found entangled structures everywhere, even in a protein data bank, so even in experimental structures. Obviously, there is not much of these structures, so in terms of the percentage of the whole subsets, obviously it's not a big one. Although we still can find structures with entanglements everywhere. Some structures contain more than one entanglement, and the record breakers we found were like 70 entanglements in one model that was predicted in one of the CASP competitions. In the experimental structures, we also found structures with more than one entanglement. And the record breaker here was a structure solved with cryo-EM, quite a poor resolution here, and it included 21 entanglements. Most of these entanglements are lassos. So if we say, okay, at least half of them are shallow lassos. So these are not big issues here. However, we also find links, which are like between 20 and 40% of all the entanglements found in predicted models. So these entanglements should be really considered as problems and should not be accepted when we predict the models of RNA. We tried to find out whether there are any classes or theoretical classes of entanglements that cannot be found in the structures either predicted or experimental. So we scanned these structures in a subset also for the types of entanglements for the presence, and we found that in the CASP predictions and in protein data bank experimental structures, any type of entanglements can be found. In the predictions submitted to RNA puzzles, we didn't observe two types of lassos with dinucleotide step as a lassoing element. Okay. Now regarding topological knots, we also scanned all these subsets, trying to find what kinds of topological knots we can find there. And here the results of analysis of CASP15 models. So we were just trying to find out what is the complexity of topological knots found in the predictions. And well, you can see that there are really, really complex topological knots in the predicted structures. Most of the knots are trefoil knots, so these are the simplest topological knots. So this is the first bar here. Over 40% of all the topological knots found, and the predictions are trefoils. But we also can see that there are other types of knots, even knots with over 11 crossings. Some knots, like around 20% of structures, were included such a complicated knots that we could not classify them. So the algorithm that we used for detecting and classification detected knots, but it was the knots were so complex that it could not simply finish classifying these knots. Okay, as for topological knots, we also know that recently, a year ago, the first topological knot was found in experimental RNA 3D structure, in a structure that was determined in high resolution. Okay, so we can say that that this is now confirmed that RNA structures can include topological knots. So the knot that was found was a trefoil, and it was found by a short bacterial RNA. So the author of this analysis of the paper postulated that this type of knot can be a conserved feature of all these types of bacterial RNAs. Okay. I mentioned already that we try to find out whether there is any correlation between entanglements, topological knots, and other features of the structure that we can evaluate or observe. So we did not find any correlation between entanglements and knots and RMSD or clash score or molecule size, but we found a correlation between pseudoknots and entanglements. So it appears that if the structure contains a pseudoknot, then sometimes the predictive models produce a structure with knot or entanglement instead of a pseudoknot. And we just computed that the probability of entanglement in a pseudoknotted structure is four times higher than a non-pseudoknotted structure. Another thing we found was that machine learning models that predict RNA 3D structure are more prone than non-ML methods to generate entanglements, including complex knots. So what you can see here on these two pie charts is the percentage of entangled versus non-entangled models generated by non-machine learning methods on the left pie chart and machine learning methods on the right pie chart. So the difference is quite, well, maybe not high, but it's observable. So we can see that over like 12% of the predictions produced by machine learning methods were entangled structures. We also found that, well, there is no direct relationship between topological knots and entanglement of structure elements in structures. So we can find structures which have topological knots but no entangled elements. We can find structures which has entangled elements but no topological knots, and we can find those that include both topological knots and entangled structure elements. Okay. And the final thing is whether we can whether there is any remedy for entanglements that we don't want to have in the predictions. So the easiest thing we can do is to run energy minimization by any tool that we have at hand. And the energy minimization actually can remove a lot of entanglements, small, let's say, shallow entanglements, shallow lassos, and also very tight links that are that were generated by the predictive model. Here is just what you can see in this figure is the analysis we did for one of the models generated by RNA Composer. So we just generated some preliminary model. We found by RNA Composer, we found that that it includes like 55 entanglements of structure elements. And then we simply ran minimization using Sienna. And with each iteration, we tried to find out whether the number of entanglements decreased or not. And in the chart here on the right, you can see how the number of entanglements was decreasing with the consecutive iterations of energy minimization. So one thing is just to use any energy minimization protocol and try to run it for a model which is entangled. And for sure, some entanglements can be removed during such a procedure. Another thing that we did was trying to run energy minimization in a kind of a way that directed just the entangled fragment. So here you can see the procedure that was proposed by Simon Pobleta. And on the short animation here and the short video, you can see where the red arrow points the fragment that actually was disentangled during this procedure. The procedure is based on SPQR that was developed by Simon and co-authors. So in this protocol, which is now which has been described in our paper, the minimization is simply run in this addressing these sites that where the entanglements were detected. Using this protocol, we're trying to find how many entanglements we can remove, and how the procedure affects any other evaluation measures that we can use to evaluate our model. So here is an example, again, of a model that was submitted to one of the CASP challenges. The model contains 7 entanglements of different types, so 4 links and 3 lassos, and we tried to disentangle them using this SPQR procedure. We succeeded totally, so all the entanglements were resolved. And what we found out also that most evaluation measures improved during the disentanglement procedure. Okay, we ran the protocol for structures, for almost 200 structures submitted to CASP and RNA puzzles models. And we just made an analysis of the disentanglement success rate to find out how good we are in disentangling the entanglements of structural elements. So what we found was that over 70% of interlaces could be resolved with the protocol, and around 40% of lassos. So we also try to find out whether the artifacts that were not disentangled were those that we considered as really problematic as artifact of modeling. And well, what we found was that over 80% of definite artifacts could be entangled with the procedure. Over 40% of the structures of the entanglements were fully resolved. Some structures that included more than one entanglement were partially resolved, and around 45%. And over 40% we failed. Okay, so these were the cases mainly containing lassos. Okay, now the take-home messages from my talk are like those. So first of all, I hope that we can pay more attention to RNA structure topology, as I think that it is as important as geometry. Most entanglements and knots in predictions are non-physical, so they are artifact, and we should not treat them as, we should not treat the models with such artifacts as good models. So ignoring topology risk in ranking such misfolds as good models. We should also be aware that machine learning based models entangle RNA, and they even do it more often than in traditional approaches. Also, AlphaFold does it, so please be careful with using this. And what's important that we can detect these artifacts, and we also can fix them. And well, I said here sometimes, but in general, if we have like shallow lassos or not very dense structure with links, we can resolve these artifacts. Okay, finally, let me just thank people who participated in these projects. So in the permanent team and the team behind RNAspider and the analysis of entanglements, there's Maciej Antczak, Mariusz Popenda, Joanna Sarzyńska, and Tomasz Żok. We also had 3 students who helped us to develop the web server for RNAspiders, and I cooperated with people from Warsaw in the analysis of topological knots. So these were Bartosz Greń and Joanna Słukowska. And as for disentanglement protocol, the work was mainly done by Simon Poblete with the help of our PhD student, Mikołaj Młynarczyk. Okay, that's all. Thank you very much for listening, and open for questions.